Right, so people wanted to know about transcription and translation. Well, they're both parts of protein synthesis. These are the 6 points in which you have to know. It's the stages of protein synthesis, which include transcription and translation. You need to know about this, and this. But first of all, it's important to understand that in protein synthesis, the DNA remains in the nucleus, but a copy, and the copy is the messenger RNA or mRNA, moves out to the cytoplasm. Okay? And after it moves out there, it's moving to, it's particularly interested in getting to the ribosomes, at which point proteins are synthesized. Okay, so we'll have a look at that in due time. The protein that's produced depends on the template that's used, the template being the DNA. And if the sequence changes, the template, if the DNA changes, then the protein will be different. Okay, so first of all, let's have a look into transcription. So first of all, DNA, that is deoxyribonucleic acid. You can refer to genetic information as nucleic acid. DNA is deoxyribonucleic acid. If it was RNA, it would just be ribonucleic acid. Okay, there's two types. In evolutionary terms, RNA is older from the early bacteria that used RNA instead of DNA. We've got both of them. We've got DNA, which is in the black here. This is DNA. And we've got the mRNA in red there. Now, DNA has an alphabet, by which I mean the base units of its language. Okay, and those are C, A, T, and G. C stands for cytosine, A stands for adenine, thymine, and guanine. Okay, you'll notice something if we look over to the RNA. What do you notice? Do you notice a little change maybe? Okay, instead of T's, we've got U's. Yeah, so we don't have any T's in the RNA. So these are all the same for RNA, same for cytosine, same for adenine, but we've got no thymine. So we're going to have uracil, and we're going to have guanine still there as well. So all the same, except for just that little change. DNA, the way it's arranged in our bodies, is as a double helix, which means it is shaped like, I'm going to draw this, like that. Okay, so it's one helix and another double helix, and there are complementary base pairs. Okay, these are the DNA bases, and then again the RNA bases are the same, except for that there is no thymine. It is uracil, which takes the place of thymine. Now, what do I mean by takes the place? Well, this black strand here and this black strand here, those are strands of DNA. Well, they don't look like a double helix. Well, yes, that's because they're undergoing transcription. And we have to unwind that helix, and we'll get to that in a minute. Before we get to that, I just want to show you what the complementary base pairs are. So you'll see here, this little C here, that is usually bound to this guanine over here. And you can see where the unraveling hasn't happened yet, lower down here, where the bases are still bound. They're very weakly bound by hydrogen bonds. Okay, and then over here we've got an A and a T, an A and a T, a C and a G, an A and a T, an A and a T, and it. You get the picture, okay? C's and G's are complementary to each other, and A's and T's are complementary to each other. So C is always with G, and A is always with T. Those are the complementary base pairs, okay? Now, obviously, as we get into looking at mRNA, if we're looking at RNA, that's DNA. If we're looking at RNA, then A binds with uracil, U, okay? And you can see that happening up here, how except all the other ones, it's exactly the same base pairing, CG, CG, GC, all that, but the A is bound to the U here, so that is the only difference. Right, so complementary base pairs. We have got let's have a little closer look at DNA. We have got what is called a pentose sugar. What does pent mean? It means 5, and that's because there's 5 carbons in that sugar. We have got a phosphate there, okay, a phosphate. We have got sugar, and then we've got our base. So let's say, for instance, that this was my adenine. So I'm going to draw a little A there, okay? That is called a nucleotide, okay? And a nucleotide is the name for the base that is attached to a sugar and a phosphate, okay? And you can think of, if you know that now that these are the bases, yeah, these things that I'm circling here, the C's, the A's, the A's, the C's, you could think of this as a sugar phosphate backbone. So when you're thinking about what the line is, you can think of the line as being a spine of sugars and phosphates, and sticking out from it is our base. Okay, so when transcription isn't happening, when it's just when the DNA is just sitting in its place and just sitting as that blueprint, that template, it is a double helix. So, excuse me, when protein synthesis isn't happening, the DNA is a double helix, that shape that we looked at before, 2 helixes around each other. But if we do want to do some protein synthesis, and the reasons that you want to do that are many, you're growing, you want to replicate the cell. Anything that needs something made will invariably involve having to make some proteins out of amino acids. And so protein synthesis is for growth, it is for repair, it is for replacing cells, okay? So anytime we're doing protein synthesis, we need to do transcription, we need to do translation, and we do that by reading the DNA. So, before we read it, we need helicase. You'll remember that suffix ase means that to break down an enzyme. So helicase breaks down the helix. So the helicase unwinds the helix, enabling a certain enzyme to come in and bind to it. It enables RNA polymerase. RNA, because that's what is binding to the DNA. Poly, which is many. Polymerase something, making many of something. So we're chaining up RNA molecules, aren't we? What happens is that all these mRNA molecules are floating around inside the nucleus, and oh look, a little G comes in and it binds to the C. Another little C comes in and binds to this G, and they're all binding to their complementary base pair, and the RNA polymerase is chaining up these mRNAs together so that they are an intact polymer. Polymer means many units together. So they're an intact unit made of many individual units there. That enabling the helicase unwinds the helix, enabling RNA polymerase to bind to a non-coding part of DNA, which is in front of the gene. Now, this will need some explaining. So, you want, in DNA, there are genes, and genes are sequences of DNA which code for a protein. Okay, they might code for hemoglobin, or they might code for, let's say, myosin that's in your muscles, a contractile protein. There are also bits that do not code for anything. So that means that they do have a use, but they are not part of the blueprint for a given protein. And one of the reasons that they exist is that you want some of these non-coding bits. You want some of these non-coding bits because it gives your enzymes a place to start and a place to stop. So let's say, for instance, that my gene is just maybe there. Let's say that my gene is just 6 little bases long, which isn't likely. Usually genes are hundreds of thousands long. But let's say it's just these 6 here, these 6 bases, and that is going to code for a protein. Now, the RNA, what it needs to do, the RNA polymerase needs to bind somewhere so that it can move along the chain that is forming and stitch the bases together. So the G is coming in and it's binding, the C is coming in and binding, and then this C is coming in and binding. Yes, they're binding to the complementary base pairs, but we need something, we need an outside enzyme to create this sugar phosphate backbone. And that's what the RNA polymerase does. So in this situation, if only these 6 bases are the ones that are coding, these are the ones that are going to code for the protein, going to make the blueprint for the protein, then these 3 here, these might be the non-coding section where the RNA polymerase would bind. So hopefully you remember that the way that DNA is arranged is that every 3 bases is called a codon. Together they bind to an anticodon, and that anticodon binds to an amino acid, but we'll get to that later. I'm just telling you why I picked 3 and factors of 3. So maybe the sequence GCC doesn't code for a protein, but it instead tells RNA polymerase to bind. And so now that RNA polymerase has bound to this GCC, it can go like that and stitch up the molecule of RNA. We've now got a large polymer of RNA that is based on the complementary base pairing, which happens because of the complementary nature of the C's to the G's, the A's to the T's, the A's to the U's in terms of the mRNA. And then we also need a point, another non-coding point, where the RNA polymerase knows, all right, it's time to stop chaining up this mRNA sequence. I've got my 6 bases in, and that will happen at what's called a stop codon. So the stop codon is doing a very similar job to what I've just explained here about a non-coding part of the DNA in front of the gene, but this one isn't going to be in front of the gene, it's going to be after the gene. So the stop codon, which is after the gene, is another non-coding part which signals the RNA polymerase to stop chaining up bases. So once the RNA polymerase has created our complementary gene, and we'll stick with the idea that it's these 6 bases, the U, U, G, U, 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 there will be another group of bases at the end of that, and that will be another non-coding section, which signals to the RNA polymerase to stop doing what you're doing, because I've got my 6 bases that I need, I've got my gene, I've got my template for my protein, so I need to stop. Otherwise it would just keep going on forever and ever, and that's no use. We don't need all the extra stuff. The protein just needs these 6 bases, we're saying. So the stop codon is after the gene. It's another non-coding part. It signals to the RNA polymerase to stop chaining up bases. Okay, so after that's happened, the newly formed RNA, mRNA, because we'll look at another type of RNA in translation, newly formed mRNA can leave the nucleus and enter the cytoplasm, on its way to a ribosome for translation, okay, which is the next step. All right. So let's have a little look back here. We have got RNA polymerase binds to non-coding DNA located in front of gene. Brilliant. RNA polymerase produces a complementary mRNA strand from the coding DNA of the gene. Excellent. And then in the following video we will look at the coding by triplets of bases or codons in mRNA for specific amino acids. I've just talked about codons a little bit, but we'll shore that up in a second in the next video, next video. So this is our transcription, and the rest of it is going to be our translation. Okay. So if you need to pause or anything like that and look back, you can do. I'm going to do my next one after school now because it's almost the end of lunch. Okay, so